Hello everyone. In today's Vanati's weekly podcast, Liquid Biopsy Part 5 episode, let's learn about the Nobel Laurette Science Wnt signaling pathways. Let's begin with bridging the gap of Wnt signaling to liquid biopsy relevant to our scientifically proven background knowledge. There is a need for much improved diagnostics and novel therapies for cancer through a detailed biological understanding of the disease. For the past 43 years, Wnt signaling has been shown to play an important role in human cell development in cancer and stem cell research community. Abnormal Wnt signaling has been implicated in many different cancer types. So far, 19 Wnt proteins have been discovered. The role of Wnt signaling was first identified in carcinogenesis and then in embryonic development. The first discovery was made by Neuschlin Volhard and Weisches in 1980 for Drosophila fruit fly wingless WG body defects. Later, in 1982 to 1984, Royal Neusch and Herald Warmers, the American Nobel Laureate, virologist and co-winner with John Michael Bishop in 1998 for their work on the origin of cancer, identified that it is not an embryogenic defect but caused by mouse mammary tumor virus activating the mammalian WG homologue INT1 gene renamed as Wnt causing breast tumor. Now we made advancement in liquid biopsy diagnostics and human clinical trials, including our pilot trial products. Wnt signaling consists of three pathways: the beta-catenin, canonical, Wnt calcium and planar cell polarity non-canonical pathways. These pathways are activated by Wnt protein ligand to frizzle receptors. When Wnt signaling is off, destruction complex labels beta-catenin for proteasomal degradation. When Wnt signaling is on, beta-catenin translocates to the nucleus, activating the transcription genes by inhibiting the destruction complex. The cell polarity, movement and survival are altered in the non-canonical planar cell polarity PCP pathway, resulting in cytoskeleton changes. The non-canonical Wnt calcium pathway signaling activates intracellular calcium signaling, reducing cell adhesion. In the next episode, I will be discussing the Wnt inhibitors clinical trial development. Stay tuned for such interesting knowledge sharing. Thank you. See you in the next podcast.